Alrighty, this is going to be renal physiology, so how we form urine, essentially. Alrighty, so there are 4 steps in renal physiology that I wrote here, which you probably can't even see, but they are glomerular filtration, tubular reabsorption, tubular secretion, and water conservation. So the first step is glomerular filtration. That is the process of most of the components of your blood, with the exception of like large anions, plasma proteins, blood, formed elements, etc., going from the glomerulus into the glomerular capsule. Now, you see this nice little membrane here? That has 3 components to it. Alrighty, so the 3 components of the membrane are 1, the endothelium of the capillary. Remember, capillaries have that endothelium, that simple squamous epithelium, so that things can go through it. This, in this case, is fenestrated, meaning there are little holes in it, big enough for some particles to go through, but not other particles. The second part is the basement membrane. Or the basal lamina. If you remember, just as a general rule, anything with an endothelium sits on top of a basal membrane. So that's the basal membrane of the glomerulus, aka the ball of yarn capillaries that you probably can't see too well because my light is reflecting on that. Alrighty, then the third layer is the slit membrane. Now, what's the slit membrane? The slit membrane is made of podocytes. Podocytes are these specialized cells that kind of have these legs. So what they do is they form the visceral layer of the glomerular capsule, so the inner layer of the glomerular capsule, and they just kind of sit there, and their little legs come and interlock like this. And those spaces between the little legs interlocking is where some stuff can go through, but other stuff can't. So essentially this allows water and small particles to get from the blood into the glomerular capsule, where we now call it filtrate. Something important to think of while we're covering this is that think about the nephron as out of the body. The filtrate is out of the body. And the blood cells, the capillaries, are in the body. That's just helpful in terminology, especially when we get to reabsorption and secretion. Alrighty, so the second step, tubular reabsorption. Now, tubular reabsorption is the movement of certain components of the filtrate, I believe it's 99%, back from the filtrate into the blood. Now, what gets reabsorbed? Materials such as Na+, K+, calcium, chloride, I forget what this is called, bicarbonate acid, I forget, and water, glucose, amino acids, all the useful stuff gets reabsorbed. Now, if we look specifically at tubular reabsorption, there are 2 routes that it can follow. One route is the transcellular route, and the other route is the paracellular route. You can't read my handwriting, but that's okay. Alright, so the transcellular route means it goes through the cytoplasm and out of the epithelial cell. I marked it here, but this is just a closer look at what's going on right here. Also, I forgot to mention this, but most tubular reabsorption happens right up here in the proximal convoluted tubule. Alrighty, so here we have the filtrate. Just use your imagination that this is filtrate. Then this is the tubular epithelial cells. So this is what's making up the tubules. Then we have the tissue fluid, interstitial fluid, intercellular fluid, whatever you want to call it. And here we have the capillary, specifically the paratubular capillary, but capillary works for now. Alrighty, so the transcellular route is going through the cell. And then the paracellular route is when it goes through the gaps between the cells. You see these little gaps there? Oops, I just erased part of the cell. Alrighty, anywho, that is when things go between the gaps in the cell. And water can leak through the gaps, and it takes dissolved substances with it. That's kind of like osmosis. If you remember, water comes out, the dissolved substances come with it, and that is called solvent drag. Alrighty, so for transcellular route, so this is for sodium and hydrogen and amino acids and glucose, just moving those all out back into the capillary because those are useful substances. We want those back in the blood. So, first of all, remember the sodium-potassium pumps? They pump K plus in and Na plus out. So we have those in the tubular epithelial cells. So we have all this Na plus, K plus, you have all this Na plus and water, glucose, amino acids. We have all that fun stuff in the cells. Now, the Na plus is going to want to go from a high area of concentration to a low area of concentration. So it's going to flow this way. And these pumps right here ensure that Na plus keeps going out and back into the blood. Now, how does it go from the filtrate into the into the tubular epithelial cells? There are two types of transport proteins at the apical surface for Na plus uptake into the tubule epithelial cells. So first of all, we have symporters. You can see I kind of made those. Symporters, that's one type of transport protein. They move Na plus and other substances like glucose and amino acids from the filtrate into the cell. So that's one way we get them into the cell. The other way are antiporters. These are kind of fun. So these are Na plus, H plus antiporters. So at the same time that Na is being, here we'll make these ones with the horizontal dashes, the Na plus, H plus antiporters. So at the same time Na plus is being transported into the cell, H plus is being transferred out. And that's really important for acid-base balance, because if you remember, H plus is an acid, and we don't want that building up in the blood too much, or else we'll have too low of a pH, and that kind of destroys the narrow window of 7.35 to 7.45 that humans need to maintain. So that's like a really functional way of getting Na plus, which we need in the blood, and H plus, which we don't necessarily want in the blood, building up in the blood at least, back into the filtrate so that it can be excreted. Now, water reabsorption. Let me just wipe off some of this stuff here. There are two kinds of water reabsorption. I'm feeling fancy, let's use blue. One, obligatory water reabsorption. Now, reabsorption, we think it's reabsorbed from outside the body, so the filtrate, remember we're pretending that this is outside the body, it back into the body, so it's reabsorbing. So we've got obligatory water reabsorption. So this is where, if you remember, the Na plus that's transported from the proximal convoluted tubule from these processes gets transported out. Well, water needs to follow. Remember, salt sucks. Salt sucks water out of it. It creates that osmotic pressure where water has to follow it. This also occurs in the descending loop of Henle, which kind of comes later in water conservation, the fourth process of renal physiology. 90% of water is reabsorbed through this obligatory water reabsorption. Now, to facultative water reabsorption. So, this is the fine-tuning of salt and water reabsorption that occurs in the final portion of the distal convoluted tubule and the collecting duct. This is also part of water conservation. So, this kind of has to do with like osmoreceptors. So the decreased water concentration in the blood is detected by osmoreceptors. Osmolarity. High osmolarity means low water concentration or high concentration of solutes. So that's detected by osmoreceptors in the hypothalamus. That causes the release of hypothalamic antidiuretic hormone. And so ADH. We have the hypothalamus. This produces ADH. Here we'll do low H2O blood. Is detected by the hypothalamus, which produces ADH, which causes the increased permeability. So we get the little slits widening, increased permeability of the membranes of the final portions of the DCT and the collecting ducts so that water can pass from these back into the blood. Alright. Now, one thing that's important to know is the transport maximum. So the renal tubule has a limit of proteins, of transport proteins that are in it. So it's important to know that thus it can only reabsorb a certain amount of solute. So we have like these transport proteins here, but then let's say we have a lot more solutes. So we can only transport the amount of proteins and the amount of solutes that we have, meaning that there are going to be some solutes left in the blood. The solute that doesn't get reabsorbed appears in the urine down here with the fancy little water droplet I made. Can you even see that? Yes, you can. And that's why, like, for diabetes, when you have such a high amount of glucose that comes from the blood into the filtrate, it can't get transported out, so it appears in urine. So that's why glycosuria happens. Now, that's enough for water and for tubular reabsorption. We're going to just erase that. Now, this is the third process involved in urine formation and renal physiology. It's kind of a fine-tuning. It's just adding materials to the filtrate from the blood, including wastes, drugs, and excess ions. So we already reabsorbed some stuff, but now we're like, okay, we don't need that. Give it back. That was too much. This helps remove certain products from the body. So remember, this is considered out of the body for our sake right now. The blood is considered in the body. So this helps remove certain waste products from the body and helps control blood pH. So things like the antiporters, the H plus Na plus antiporters that were important for getting Na plus out of the cell in reabsorption. That's like the biggest example I have is H plus going back from the blood into the cell. Now, water conservation. This one's kind of tricky. So I'm going to erase our lovely little thing over here, which is kind of sad because I liked drawing that. It makes me look smarter when I draw little diagrams like that. Alrighty, so water conservation. We want to make sure that the kidneys don't excrete too much water, because otherwise, well, we need water to live, we need water to keep our blood pressure up, etc. So the concentration of urine depends on the hydration level of the body. Now, how do we get water to stay in here and not get reabsorbed? Well, there are two processes we have for this. So we have one, that counter current multiplier, and two, the counter current exchange system. Alrighty, you probably can't read that. Alright, so the counter current multiplier really revolves around the nephron itself, while the counter current exchange system has to do more with the capillary, which in this case is the vasa recta. We'll get into that when we get into the counter exchange system in more detail. So the counter current multiplier. Counter current means two tubes running in different directions. So we have the downward tube here, right next to the upward tube right there. Counter current. Simple. And then multiplier means, kind of refers to the solute pumps multiplying the solute in the medulla. So, as a general rule, it's important to concentrate urine so that the renal medulla, this is the medulla of the kidney, out here is kind of the renal cortex, but in here is the medulla. Alrighty, so we want to make sure that that stays salty. So we'll use orange for that. I'm feeling cute. Alrighty, so we have the salty medulla. Now, the ascending loop, the going up loop of the loop of Henle, is impermeable to water, but permeable to NaCl, meaning it pumps out NaCl. So in this portion, we are pumping out of the blood and out of the filtrate NaCl. And that keeps that keeps the medulla nice and salty. Now, the descending limb is permeable to water, but not NaCl. Therefore, water, as salt sucks, we have all this salt in the medulla. We want to get water out of here into the medulla. Now, H2O, H2O, H2O. Alrighty. So, the filtrate in this part, as you can see, as more water leaves, that means we get more salt in the filtrate. As we go down, it gets increasingly saltier. And down here, and then the loop of Henle, it gets the saltiest. Now, as we go back up, now we have all this water in the medulla, but we don't really want that. We want to make sure that NaCl is still, we want to make sure that the medulla is still salty. So, up here, aforementioned, we have pumps pumping NaCl out into the medulla. Now the question is, the large amount of water that's being reabsorbed into, we must reabsorb the water into the blood. But how do we do that without reabsorbing the salts that we need to maintain the salty medulla? So if you remember, the water's going to want to stay where it's nice and salty, but we need the water to get back into the blood. Okay, how do we do that? Countercurrent exchange system. So, blood flows down this way, up this way. So, countercurrent. In the downward segment, so over here, water diffuses out of the blood and NaCl goes in. I forgot to use orange for NaCl, but that's okay. So, this means that we have really salty blood down here, which kind of, we don't want that. We want more salt in here. More salt to maintain that salty medulla. Now, in the upward segment, H2O goes back in because it sees all this salt in the blood, and so it wants to go back in. And that's happening over here as well. It's just H2O goes in. And NaCl goes out. Oops. NaCl. So that makes it get increasingly less salty. So now we have the water from here reabsorbed into the blood, which we want because water is an important nutrient, and we have the NaCl leaving the blood and going back into the medulla to keep it nice and salty. I think that is it. Those were the 4 processes of renal physiology. So we have water reabsorbed, the water we need reabsorbed, and then we have the Na plus out here in the medulla, and things are cool. The filtrate goes down, becomes urine. Now, specifically about the concentration of urine. So urine is dilute. It doesn't have many solutes when compared to the water concentration when you are well hydrated. When it's nice and dilute, meaning there's like more water than solutes when you're hydrated, ADH, so that antidiuretic hormone, is not secreted. So the this collecting duct up here reabsorbs more water. Reabsorbs more salt without reabsorbing water, so that the urine is more diluted. So more salt goes back into here while the water is chilling. Because we don't really need all that. We're well hydrated. We already have enough water in the bloodstream. Now, if urine is concentrated, meaning there are a lot of solutes in it compared to water amount, that means that you're dehydrated. You want to hold on to that water. So down here you have a lot more solutes compared to water. So, antidiuretic hormone, which we mentioned up here, is secreted, and that makes the collecting duct down here reabsorb more water. So water goes out here. If you remember, when there's low H2O in the blood, the hypothalamus senses it, secretes ADH, and that increases permeability of the collecting duct, which lets water go out and get reabsorbed back into the blood. This is made possible due to the hyperosmotic medulla resulting from the countercurrent multiplier. And so the vasa recta, so this, is arranged as the countercurrent exchange system so that water can be removed from the interstitial fluid back into the blood without disturbing the concentration gradient. Okay. That is it. Anywho, I kind of lost my brain there a lot. But, womp womp. Have fun.